quickly, here you are at the CHU de Québec, Centre de recherche du Centre hospitalier universitaire de Québec, which is it's the largest francophone research center in health in Canada. While in Montreal, there's several research centers or French-speaking research centers and also English-speaking Anglophone and Francophone research centers. But in terms of the number of researchers and students in a Francophone institution, the CHU de Québec is the largest one. We are the 8th in Canada among the research hospitals. It's the 8th in Canada in terms of research intensity. And the main area of excellence here, I would say, are cancer research, neurosciences, virus and viral, the development of vaccines, for instance. So there are 8 research axes at CHU de Québec, and we belong to the cancer oncology axis with a lot of links with regenerative medicine. So I've been in the regenerative medicine axis for a long time, now mainly active in the oncology axis. So the main objective of our laboratory is to develop innovative biomedical devices, technology and processes for the diagnostics and therapies in oncology. And we align our main research activities according to three pillars: nanomaterials synthesis, the strong expertise in nanomaterials characterization, and also a flexible capacity for functional assessment in imaging and medical physics. So we always try to master, I would say, a very focused series of nanomaterials synthesis, mainly iron oxide nanoparticles and gold nanoparticles. My students know very well about this. Then a capacity to analyze nanomaterials in terms of scanning electron microscopy, different types of spectroscopies, as well as nanoparticle size characterization and polymer characterization as well. And we always keep a very close eye on the functions of these materials because we have many ties with the medical physics and radiology field, so many collaborations with medical physics and with radiologists. So the main specialty of the lab is the capacity to visualize a biomaterial in vivo. So when you want to visualize with MRI, with CT, or with positron emission tomography, what happens to materials that you implant in vivo, this is what we do in the lab. So it starts with the synthesis of a nanoparticle, characterization of the material, and we also run a small animal imaging platform with PET, MRI, CT, mainly PET, MRI, and CT, and we do some ultrasound as well. Recently, we've started a new research project where there's a little bit more of ultrasound that we used to do. And it calls for the use of a complex series of equipment. Of course, we have a scanning electron microscope in the vicinity here. We have a capacity to visualize nanoparticles, iron oxide and gold nanoparticles with transmission electron microscopy in partnership with my colleague Sachiko Sato, who owns the TEM. So these two instruments are very, very important for the characterization of nanomaterials, and we do use the nanomaterials in 3D printing processes. And of course, the small animal imaging platform with positron emission tomography and MRI, which we have always used, since the beginning of the lab 10 years ago, we have always used these small animal imaging equipment to test the nanomaterials designed in the lab by the chemistry students. So probes and contrast media for biomedical imaging is an important aspect of what we do. We are not that much involved in optical fluorescence, optical imaging, including fluorescence probe. We do use fluorescence, but it is not the main aspect of our research. I would say that MRI, CT, and PET are really by far the projects or the modalities that invariably involve our finality. So in order to know what it takes in terms of contrast or product in order to visualize an organ or an implant in a specific organ, we really need to know the magnetic properties of nanoparticles. So we have an expertise on how to design, select, purify, blend with polymers, iron oxide nanoparticles, manganese-based products or gadolinium-based products, depending on the finality of the functional application. CT, of course, is a major tool. We didn't use that much CT before we started the 3D printing laboratory about 7 years ago. And we have been using the CT much more for validating the precision of the 3D prints. So now I would say that we use the CT scan much more for 3D printing than compared with what we used to do before for the small animal. So CT is kind of a renewed functionality in the lab. So you 3D print an object according to numeric prints, and in the end, you end up with an object that is not necessarily as perfect as you wish, which is one of the main challenges in 3D printing. So you go back to CT and you assess the precision of the geometry with CT, and then you try to change the numeric plans in order to facilitate the printing of an object that is as close as possible to what you wish. So this iterative process is facilitated in the lab because we have a CT scanner. And also, I will not touch today that much about positron emission tomography, but it is also an important aspect of what we do. So I've selected, I will keep it very short to about 10 minutes, but I will go straight into the applications where 3D printing have been very important to us in the last 5 years. First, the development of 3D printed brachytherapy devices. I will explain rapidly what brachytherapy is. Radio-opaque filaments for the printing of medical physics and radiological objects, and the development of gold nanoparticle eluting device surfaces. And this application calls for a bioprinter because we develop gold nanoparticles that we use as an enhancer of radiation therapy for medical physics. And instead of injecting gold nanoparticles directly in the small animal, we have been using more and more hydrogels. And hydrogels applied at the surface of a brachytherapy device. Brachytherapy devices are radioactive implants that are inserted in the body for radiotherapeutic treatments. So these projects were initiated about 5 or 6 years ago when we implanted the 3D printing lab. And Théophras Lescaut, who's now a research associate in my group, he was the one to pioneer the hard polymer printing infrastructure was initiated by Théophras, and there were also other students for the bioprinting laboratory. So the main objective of his project was to develop this kind of object here, and this is called an episcleral plaque for brachytherapy. This is a plaque that is more or less the size of a loonie, of a dollar, and it is sutured at the surface of the eye by surgery, and it is placed for about not more than 4 days and removed by surgery. And this object is radioactive, and the main use of that object is for the treatment of cancer, because eye cancer is a relatively rare disease, but it has been increasing because of UV exposure and so on, and aging population. So the number of cases of eye cancer have increased, and in Canada, episcleral plaque is probably the most widely used treatment for a certain class of that type of cancer. And the objects are not necessarily personalized. Actually, they are not personalized at the moment. They are very standardized. So the size, the curvature of these objects are always the same, with some variation, but very limited variation. So at the core of the project, we were looking for a possibility to extract the anatomical data from the patient. And anatomical data is the size of the eye, the curvature of the eye, and the size of the tumor and where the tumor is located with respect to the optical nerve or the retina and so on. And this is where personalization of this type of treatment is interesting. And for this, we needed to demonstrate the possibility to 3D print a plaque, which usually is made of titanium and sometimes hard polymers, but mainly titanium. And that was one of the first time that we tried to an episcleral plaque and a brachytherapy device was 3D printed. So basically, you have here the traditional type of plaque. Radioactive seeds are placed at the surface. They line the surface of that plaque, and the plaque is sutured at the side of the eye, and it is left there for about 4 days. And removed by surgery, placed by surgery, removed by surgery. And this is the very, very precise and very specific geometrical configuration that you need to achieve in order to be conformed to the regulations of the radiotherapy. So this type of protocol was developed for a machine type of metal device, and we needed to transfer it to a 3D printed polymer. So we developed that flow of development. So from a CAD, a computer assisted design, then you adapt the CAD design with different types of features that are specific to 3D printing. Like for instance, the brim, everything that is purple here refers to a very fine layer of polymer that is required to stick your object at the surface of the plate. You will see pragmatically this afternoon what it means in terms of in front of the equipment. So that is the brim. You also have sometimes a feature like this here that is called a sacrificial tower. A sacrificial tower for certain types of polymers is absolutely necessary. It is wasted, but it is necessary to introduce these kind of little strategies in the 3D printing procedures in order to let a polymer that needs to cool down or that needs to be maintained at a certain temperature after the second pass or the third pass need to wait a little bit. So that sacrificial tower is a kind of strategy for the filament to still continue its melt while avoiding to go back to one of the prints before it reaches a certain critical temperature. So these little strategies are not something that was really evident at the beginning of 3D printing as a new, I would say the 3D printing about 10 years ago was relatively new in different types of fields, including biomedical engineering. And then we researchers involved in 3D printing procedures develop these little strategies here like brim and sacrificial tower and everyone. And I'm mentioning these things, they are very, very technical, but are very important. If you want to master fully, if you really want to 3D print efficiently and reach your goals, and not in 3 years, but in 6 months or limited amount of time, it's important to get to know these little tricks and to talk to a research associate or a student who has been really involved into these technical procedures. And then there's the post-processing. There's different ways to work on the print just after it is 3D printed. Just after 3D printing, it's not perfect, of course, because there's the brim, there's a sacrificial tower, and there's different types of features that you need to, and you need to polish because the 3D printed device is not perfect. It's not smooth. Usually there's a kind of variation at the surface and velocities. So post-processing, depending on your requirement, will be necessary. And then you go to the CT scanner and segmentation, followed by some comparative analysis between the CT scanner results and the CAD plan. So if you are really, really far away, you are more in the red or in the dark yellow and orange. And if your results are good and within the limits of the geometry, within the requirements of the geometry, you should see some green pixels. So we have developed these kind of geometrical comparison tools, which are extremely useful for what we do in terms of conformation. And so we want to reach a certain level of geometrical compliance, because if it is not compliant with the regulations of medical physics protocols, it's not going to be applied in the patient. So this is an important aspect of what we do. And then we develop some workflows to place the radioactivity in certain patterns of radioactivity inside of the implants. First, for the assessment of radioactivity placement in the 3D printed parts, we started to use 2D radiographic films, and it was pretty clear very early on that 2D radiographies were not a good tool for our applications. And this sort of initiated 3 projects here in the room related to dosimetric hydrogel. So instead of using a radiographic film, a 2D object, we use dosimetric hydrogel. So we have not invented the dosimetric hydrogels, but we have and we being Marduk, my student over there, and other students in the lab, they have worked really hard to adapt these gels to the realities of the radioactive 3D printed devices. And we have a collaboration with Hôpital surgeons from surgeon Claudine Delrive from Hôpital Saint-Sacrement, who is an expert in the placement of these plaques on the eye. And we have very good collaboration with her. She tests manually each one of the objects on real human eyes, eyes from the donors, and we get pretty straightforward feedback about whether or not the prehension is good, if she can manipulate with the tweezers and so on. So the feedback from a surgeon is absolutely necessary, because if the polymer is slippery, if it doesn't work pretty well in the surgery room, the technology will never be adapted by the people working on these in the OR. So quickly, so that was for the brachytherapy. So now there's many projects related to the development of different types of brachytherapy devices, not just the eye, but also cervix cancer and also different types of cancer that can be treated traditionally by brachytherapy. I will not cover all of them. I will jump into the development of radio-opaque filament, because it is really logical to go to that direction. And this is a presentation from my student Soheb Zekraoui, and in collaboration with Marduk Akbari here, they went to the World Congress of Brachytherapy this summer in Washington, D.C. And they presented the latest results of the 3D printed plaque. And one important component of the 3D printed plaque for eye cancer therapy is the new filament that we've been using. And you see, the color of the polymer here. The color of the polymer that you saw a few slides before is the natural color of polyether ketone. It is a pinkish colored polymer. Why we're using PEEK, a very hard polymer? That's because first, it is hard, and also it is really resistant to chemical degradation. And in implantable polymers, you want a polymer that will stay and remain in the body for long times, for long-term applications, but also in a state that doesn't degrade and does not elute any residual polymers or residual unpolymerized components. So PEEK is really good with that, but it is not radio opaque. And a brachytherapy device must shield the radiation at least in one direction. For instance, when you place an episcleral plaque at the surface of the eye, you don't want your eyelid to be completely cooked by the radiation. You really want the radiation to be projected towards the inner side of the eye where the tumor is. So you want to block the radiation. So by using a polymer that doesn't block radiation, you lose a very, very significant function of the traditional plaque. So very early in the project, we realized that we really needed to develop radio-opaque polymers for printing radio-opaque plaques. Instead of machining metal parts, we realized that we needed to 3D print polymers that have a very, very high content in metal. So that's why we ended up developing filaments, filaments that with PEEK that contain a very high fraction of tungsten. We can blend gold, of course, it's just more expensive. Tungsten is relatively biocompatible. It is encapsulated by the polymer. We expect that it will not leach that many tungsten ions, which are not necessarily very, very toxic. So this is a toxicity that can be controlled pretty well. And there are other metals that we can blend into these polymers. So we developed these filaments with a very high content of metal. They were assessed by CT imaging and different types of modalities to demonstrate that they really do block the photons that are used as the radioactive component. The CAD model, the 3D printed plaque, the 3D printing strategy was developed, followed by a development of a dedicated bench for drilling these little holes that are used to place the radioactivity in the plaque. And this is a strong contribution from Théophras Lescaut, research associate in my lab. So we've developed this little machine tool that is very close to the I would say the watchmaking industry in Switzerland is doing these kind of things. So we clearly inspired ourselves from the watchmaking industry. And why was it important? That's because the regulatory approvals for the medical physics workflow requires to drill a certain number of holes and to place the radiation at certain places. But the personalization is an important aspect of what we do, because we can decide what activity we place in which one of these little holes. So this is one of the aspects of the personalized plaques that we have developed. And again, CT scanning is used to demonstrate the quality of these little drills. These little holes are placed at very specific places in the plaque, and it is assessed by CT to demonstrate the correlation between the CAD plan and what we obtain in the end. So we use, once again, CT scan very frequently in that workflow, because it will be important when we go to the patient to clearly demonstrate the quality of the print, the quality of the geometrical positioning. And then here is the little capsule that is used for the dosimetric gel. So I mentioned you earlier that we have developed a project where focused on the development of dosimetric gels. And dosimetric gels are, instead of using 2D radiographies, we use a gel that collects the radiation, or so radiation prints a polymerization effect inside of the gel, and that polymerization is readable in MRI. So we use MRI as a kind of camera to reveal how radiation has printed or deposited energy into the gels. So rapidly 3D printed workflow and placement of radioactivity, assessment with CT scanning for the precision of the geometry and the place of these little dots, these little holes, followed by placement of this radioactive plaque on top of the dosimetric gel. Here you have Marduk. And these are the images that we collect after we go to the MRI. So we go to the MRI, we let the radioactive implant on the gel, the dosimetric gel, for a certain number of hours, usually around 48 hours. And then the little sarcophage or little capsule that contains the hydrogel is inserted into a MRI, and the MRI collect the images, and we reconstruct in 3D the dose profile. So where the energy has deposited at a very high level, you have the red, and where there's a low deposition of energy far from the radioactive plaque, it is far. So this line here represents the contours of the eye, and eventually the tumor will be here. So we try to shape in 3D the dose profile in order to make it as close as possible to the geometry of the tumor. And that's the objective of the medical physicist. And that's also our objective in terms of biomedical engineering of the radioactive implant. And so there's a whole strategy for positioning carefully the dose profile, the dose contours around the contours of the tumor. And then we compare with the traditional plaque, where you see that the traditional plaque is much more wide, much more vast. So these lines, these dotted lines here that represent the traditional way of treating the tumor, reflect one of the very strong disadvantages of the current technology, which is you over-irradiate the sclera. So that the white part of the eye on both sides of the tumor gets irradiated too much. And if the optical nerve is too close, well, you grill the optical nerve, and it will have an impact on the vision of the patient, of course. So we can demonstrate that the 3D printed plaque and the specific positioning of radioactivity holes has a great impact on the capacity to get that dose profile to a narrower level. So you see here a combination of hard material 3D printing plus the use of a dosimetric gel that can also eventually be 3D printed. Not in this specific case, it was not. Eventually, it can become under certain consideration. And there's also the filament fabrication that contains a very high amount of metal that we use for the fabrication of brachytherapy devices. You have here three very representative examples of what we do in the lab, and that will be illustrated to you this afternoon during the workshop. So I will go really fast on the last part, just a kind of teaser. We also work a lot with gold nanoparticles as radiosensitizers in radiotherapy. So gold nanoparticles located in the vicinity or distributed in a specific tumor or in a specific organ that you place in a beam of radiation are susceptible to accelerate or to enhance the dose, the dose achieved in the organ. So this is called a radiosensitizer. And we have performed a project as a proof of concept for the use of gold nanoparticles as a radiosensitizer for cervix cancer therapy. And my former student, Maria Kiseleva, who is now working at Reximage in Montreal, a radioisotope company, has been working on a quite complex combination of hard polymer, hydrogel, gold nanoparticles in the hydrogel for an elution capacity of a device to deliver gold nanoparticles at the surface of the cervix wall where a tumor is located in the case of cervix cancer. So she has developed a type of material that, a hybrid material that contains gold nanoparticles with pluronic, alginate. Why pluronic and alginate? That's because these two polymers are widely accepted by the FDA for topical applications and also for injectable devices. In combination with gold nanoparticles, we developed this kind of polymer that can be used for a specific treatment that is, well, probably cervix cancer. About 20% of cervix cancer cases are treated by this kind of procedure where you combine chemotherapy and radiotherapy by external beam or by brachytherapy. So brachytherapy is a technology that is very, very interesting for the treatment of cervix cancer, but that it is a technology also that could be accelerated by the use of gold nanoparticles, provided you inject the gold nanoparticles topically and locally and not through intravenous injection. If you inject gold nanoparticles through intravenous injection, there's high chances that you get a lot of gold in the liver, the spleen, the kidneys, and that's not good for the body. It's not necessary for the patient. So that's why a topical application by a gel for the delivery of gold nanoparticles after the insertion of a radioactive source inside of the patient is a strategy that can have a chance to make it to the clinics because it avoids a lot of toxicity related to nanoparticles. And I will go quickly on the 3D printing aspect. So one important aspect of this project was to be able to manipulate the gel. To place the gel at the surface of the device was very important to us, not only because of the geometry, but also to the dose. With the syringe, bioprinting actually, and you will see it this afternoon in the bioprinting workshop, it's basically a syringe with a controlled mechanism. And you need a certain viscosity in order to get in control of the geometry of material that is deposited at the surface. And if you get the right viscosity and a polymer that gelifies adequately at the surface in the Petri dish or in the surface of where you're working, like a radioactive device, then you can also control the dose of the drug or the nanoparticle that is placed at the surface. So here in this case, bioprinting was not that much used for the geometry, but it was rather used for the dose. So the brachytherapist needs the calculations from the medical physicist in order to make sure that the treatment is compliant. But if you have gold nanoparticles or a drug in combination to gold nanoparticles, then the pharmacist is the pharmacy aspect is important. The pharmacologist expertise is important. So as a matter of dose control, 3D printing is also interesting. And if you look at the current trend in pharmacology, there's more and more papers of 3D printing in the field of pharmacology. There's a very interesting increase in the number of papers and possibilities. So for the sake of time, I will stop here. So we characterize the materials. So here you have an example of the 3D printing process with the cell link by printer that we have in the lab. You will see it in action this afternoon. and cell culture experiments for the biocompatibility of the product, injection in a small animal, flank tumor insertions, and a little device here is made with the hard peak 3D printed procedure that we developed for the epithelial plaque therapy. And you see here the gel, the dosimetric, not the dosimetric gel, but the gold eluting gel that is placed inside of the peak epithelial plaque. And that peak epithelial plaque was adapted for a mouse model. So we have tested xenograft tumors, human tumors, grafted on the flank of the small animal, and we have treated them with the eye plaques adapted for the animal model and used as a support for the gold nanoparticle eluting gel. So it's a combination of different techniques and different technologies that have find connections and intersection one with the other. So in conclusion, I think my main take-home messages here is it's a representation of what we do in the lab in terms of 3D printing. So 3D printing is not the final objective of the lab. It's really a tool. It's a tool, and it's a good tool. I think I like to have access to this tool, but certain of my students, like for instance, Raquel, can tell you that sometimes 3D printing is not the solution. Sometimes molding is the solution, right? For the eye model, echographic eye model that you've been using. 3D printing, biogel, 3D printing is not the right solution, is not the right approach. So you need to test these different approaches by yourself in order to know if it is good, if it is not. There's always a lot of work related to the development of the 3D printing workflow. So do not hesitate to contact us and to discuss about your challenges, and we'll be very happy to help you. So thank you very much. I will take a few questions maybe.